How are you today? Busy day today? Okay, so good morning. My name is Miguel Toribio Mateas, and I'm the chairman of BANT. For those of you who don't know me, I think most of you are members of BANT anyway. Just a few bits and pieces of housekeeping today. We've got a gut day ahead of us and modulating the microbiome. You know that you'll be receiving your CPD certificates at the end of the day electronically. You've got a delegate pack that you can download if you want to make notes in your computer, but then you've got your print pack as well. And we're going to start the first presentation in a few moments, and the day will finish at 4:30. And from about 4, we'll have the chance to do questions and answers. So there won't be questions at the end of each of the talks, but at the end of the day. So if you have any questions that may pop up as we go along, if you write them in a piece of paper and hand them over to anybody hanging around the room, you've got quite a few facilitators of the CAMP conferences team hanging around, so just hand them over and we can see you at the end of the day. And I'm just going to introduce Ben Brown. So Ben is a naturopath, science writer and speaker. He's on the advisory board and faculty of the UK College of Nutrition and Health, BCNH. I'm sure there are a few graduates and students of BCNH today here. And he lectures at various educational institutions at university settings. Ben regularly speaks at major conferences. He was telling me earlier that it's his third speaking commitment this week, so he's a busy speaker. And throughout his career, he has had extensive clinical experience maintaining a private practice in Australia and in London. He's worked in nutraceutical research and development, in product development for a number of leading global companies, and served on advisory boards for integrative medical clinics and wellness services around the world. He's an established science writer and frequently contributes to peer-reviewed scientific journals. He's an associate editor of CAMP magazine. You've got copies of CAMP magazine as well to pick up from upstairs, and an associate editor, and an author of the Digestive Health Solution. Ben's qualifications are in naturopathic medicine and integrative health, and I've known Ben for a number of years now. He was one of those that when I was still studying, I was always looking forward to seeing in lectures, and that hasn't changed, so I'm looking forward to listening to him today again. So welcome to the stage, and thanks for being with us today.
It was a pleasure.
Cheers. Thanks.
Enjoy.
Very nice. Okay, so I'm going to start this one with a question. Have you ever seen a patient that has been through some sort of significant gastrointestinal stress? Maybe it was an infection while they were abroad, maybe they had a previous illness and they went through a strong course of antibiotics and they just don't recover. So they have a very sensitive gut, it's very unstable, and they're prone to dysbiosis or digestive health related illness. Another question, have you ever seen a patient who you've prescribed probiotics to that doesn't respond in the way that's predictable? Maybe they get worse on a probiotic, maybe it doesn't do anything at all. So what I'm going to try and do, at least conceptually over the next hour roughly, is try and shed a little bit of light on why that might happen. So the theme of our conversation now is dietary-induced dysbiosis, its connection to inflammatory disease, and we're also going to look at that through the lens of what we can do about it. So our understanding of this thing we call dysbiosis has been revolutionized like just in the last month. I am just amazed at how quickly our understanding is changing around this topic called dysbiosis on like a daily basis. It's just flabbergasting. And I'm sure, like a lot of you, I keep an eye on the scientific literature, and I'm just really interested in this topic because, as we all know, really the best place to change this dysbiotic picture is with nutrition. So we are the most important, I think, healthcare providers in terms of changing this scenario, but we'll get to that. So our understanding is revolutionary. So if we look at the history of dysbiosis, we didn't even know there was a microbiome about 60 years ago. It was a bit of a shock to discover that there was this ecosystem living in our digestive tract. In fact, it was previously thought we were relatively sterile. And then when we started to introduce culturing techniques in the 1960s, some of the first bacteria we were able to culture was E. coli. So we thought originally that actually E. coli was one of the dominant strains that made up the human microbiota composition. And then culture techniques advanced a little bit. Then we identified about 300 different strains about 10, 20 years later, and we thought that was pretty sophisticated. And now we have these DNA sort of shotgun sequencing techniques that have just opened up a whole world. And we now know that there are thousands of different species in our digestive tract, and each of us are incredibly individual. So this old idea that I was taught in college, that dysbiosis is just an imbalance of good and bad bacteria, is kind of defunct now. So we're not talking about species anymore, because each of us are incredibly individual when it comes to our microbiome. We don't really have a load in common. About a third of our genetic material in the gut is shared across human populations, but the rest of it is highly individual. So what we're starting to realize now is that this species definition of dysbiosis is being superseded by a metabolomic or a metabolic definition of dysbiosis. So it's not so much the type of bacteria you have, but it's the way that they're functioning and interacting with your body. So a lot of what we're doing clinically is just guesswork. We don't really know what's going on down there. But what I'm going to try and do over the next hour is shed a little bit of light on some of the more recent understandings, especially with how they intersect with nutrition. And that's where it gets really interesting for us, because it gives us some clues on what we could be doing to help patients. So that's where we're going to go. I guess the core question we're going to ask is what's changing the gut microbiome and its metabolic function? And I'm going to propose that there are two key things we should be looking at clinically. Firstly is diet, is by far the most important. If you look at, things like prebiotics and probiotics have an effect, but it appears to be quite limited. Probiotics, for example, have a very transient effect. Within a week, most of them you can't detect them anymore in a stool, or in samples. And their effects on the overall colonic ecology are marginal, if anything. So they seem to be working mostly in the small intestine. And if you look at diet, however, that seems to have a tremendous impact. So the first thing is diet, and I think that is probably first and foremost in terms of changing gut ecology. The second thing that I think is little talked about is actually our physical function and state of health. So the way our body is functioning and the capacity and health of our digestive system, I think has a major influence on our digestive ecology. So I'm going to expand a little bit on that as we move forward as well. But I'm predominantly going to focus on dietary influences. So before we get into the news to use stuff, what I'd like to do is lay a little bit of a foundation, and then we'll jump into some of the things in our diet that we know are impacting the microbiome, and then we'll flip that around into some clinical stuff, and we'll look at some research that gives us some clues on what we could do about it. But in the first part, laying that foundation, I want to point out a couple of basic things that we know for sure about the microbiome. And one of them is that we have a microcosm or a microecology living inside our digestive tract. It's all over our skin and in our mouth and esophagus and even stomach, but particularly rich in our colon or a large bowel. That's where the preponderance of these bacteria are. And the fascinating thing about these bacteria is they are shared across the species that live throughout our environment as well. And I think this sounds a little bit esoteric, that these bacteria living inside us are also living in the ocean and on trees and in forests and in the soil and even in the air. But there's a really important message in that I'll come back to as we move on. We are connected to the environment around us, and that could be having a big impact on our digestive health, strangely enough. So So there is a microcosm that lives within us. There are about 52 phyla of microbes or bacterial species that are generally accepted on the Earth. About 5 to 7 of those live in humans. And amongst those 5 to 7 phyla, we have about 1,000 different species that we're aware of, living in the, human ecology. What's interesting, as a lot of you already know about this gut microbiome, is that it has about 150 times the genetic material of our own body. So as I heard David Perlmutter put it once, it seems that we've uploaded most of our genome to the cloud. And the cloud is our gut microbiome, and there is actually a beautiful analogy in that we're actually using our microbiome to draw on facultative functions that we don't otherwise have in our physiology. So they have digestive capacity, they can adapt quickly to the environment, they adapt quickly to dietary changes, more than our body can in a very short period of time. Within a day, within a meal, your microbiome is starting to adapt to that environment that we're exposed to. So we're drawing on that genetic resource, which is interesting. The second thing I just want to highlight a few elements within that ecology itself. And that is that, the second thing I want to point out is that beyond that microecology is that we're highly individual. So I'm not going to read out this, it will just bore everyone, but that's in there for your reference, and I can't black out slides on this. But the point is that we're highly individual. We share about a third of that genetic material across the microbiome, but the rest of it is highly variable. So from person to person, there are very unique species, and we call this the variable microbiome. So there's collectively a kind of shared human phyla or microbiome, but then there's a high degree of individual variability. So this is really interesting. And also that will change dynamically depending on your environment. So the food that you're eating and the environment you live in, including your geography, for example, will have a massive impact on your microbiome. So it's highly individual and highly variable. And this idea that there's good and bad bugs is a little bit defunct. When we look at the dynamics of the microbiome, that's where it gets really interesting. And our understanding of this has really evolved a lot in the last few years. We now understand that rather than there being dysbiosis and homeostasis, actually there are different set points. So we can move dynamically across different states of homeostasis, which is a lot like the stress response, isn't it? When you're under a lot of pressure, your body adapts and it maintains a stable state, but you're under pressure and you become less resilient, although you're holding it together. So it's a different and dynamic state of homeostasis, analogous to allostasis in stress research. So what you're looking at here is a dynamic picture of exactly what's going on, and I'll step you through this. So up here, we start with an infant. So an infant is born relatively sterile. So their gut ecology is not very well developed, and the first 1,000 years of life, so 2 to 3 years of life, are where our gut ecology is established. And actually, once that is established, it becomes fairly immutable for the rest of our adulthood. So it's hard to change that early establishment of ecology. So we have what we call primary succession. A lot of this has analogies to environmental research. So primary succession in environmental research is where you have a sort of sterile environment. So I went to Iceland recently, which is an amazing environment. It's so weird. But one of the weird things about Iceland is they've had a lot of volcanic activity. So they have areas along the coastline, for example, where there's just rocks. So the ecology hasn't had time to thrive in this environment and develop and create an ecosystem. So that's exactly what's happened in early life, is that we have a blank slate, so to speak, and these species haven't established yet, but they establish in the first few years of life, and we end up with our adult microbiome. And this is very, very stable, it seems. It fluctuates a bit, but there's a strong element of stability across this microbiome. Now, what's interesting, as you can see here, a so-called healthy state then becomes resistant to disease. So it is perhaps resistant to colonization by unhealthy microbes or pathogens. But of course, it can be perturbed. These are all maintained by positive and negative feedback mechanisms, we think. So it can be perturbed. So for example, if you took some antibiotics, that would change your gut ecology. We know that degrades a lot of the species and flora. And, in some cases we recover. We take a course of antibiotics and we bounce back and our gut health returns. But that doesn't always happen, right? And you've seen it. We know that antibiotics are often a precursor to functional gastrointestinal disorders because it disrupts the microbiome, and then we end up with basically a different degraded stable state. And, look at someone with irritable bowel syndrome. They can have IBS for years and decades even. So what's happened is this initial disruptive event has changed their microbiome to a different set point, and now they're remaining stable within that different set point. But it's causing a lot of disease and dysfunction, so they're here. So it's a degraded stable state. So of course there are other dynamics that are sort of defined here, including this push of stresses into a different threshold. So it may not be just an acute disruption, but chronic stress and pressure over time. So that may resolve, or it may just progress into an alternate stable state of disease. And this then could be difficult to recover. So it may be resistant to colonization with healthy microbes, and difficult to return to health. As a lot of us experienced, it seems that some people just become dependent on probiotics, or perhaps, be a good candidate for fecal microbiota transplantation, which is really resetting the whole microbiome. So this is interesting. This is the dynamics of the human microbiome and these different set points that exist. The other thing I want to point out, beyond this dynamic individuality or inter-individuality, is that your microbiome is shaped by you and your environment. So it's shaped by your physiological state, and we'll look at examples of how that occurs, particularly in the context of inflammation, but it's also shaped by environmental factors. And this was a beautiful study that was published relatively recently. This is in April this year in the journal Science. Cool study to have a look at. Basically, it was a study on about 1,100, I think, healthy Dutch individuals, or relatively healthy Dutch individuals, and they did fecal samples and looked at the DNA in these to sort of categorize the microbiota, but they also collected a lot of other data, including health markers and blood work and dietary intake and things, and then tried to correlate different variables with variations in the microbiome. And basically what they were able to show is that there's a high degree of influence from different environmental factors and physiological factors. So they were able to identify about 60 dietary modifiers of the microbiome, or at least that we think are modifying it. They were able to identify about 19 medications, including common ones, not antibiotics, that you may not have thought of. And there were also important physiological modifiers as well. So there were strong correlations with different blood work and things, which is interesting, and we'll dig a bit deeper into that. But the point is, just generally speaking, at a top level view, is that your microbiome is modulated by your environment. In particular, your diet. So this then brings us to the topic of dysbiosis. So what is dysbiosis? Well, by definition, it's basically a change in the bacterial species or their function that is implicated in the development of disease. That's kind of the short definition. So what you're looking at here in this image is the gut microbiota magnified 2,500 times. And really what you want in a healthy microbiome is a lot of diversity. Like any good sort of healthy ecosystem, you want a high number of diverse sort of species that perform different functions. This improves the resilience and less disruption and loss of diversity. So interestingly, what you're looking at here is Giardia lamblia, which is one of the main causes of this loss of diversity, particularly in post-infectious IBS. It gets in there, produces a lot of toxins, wipes out the gut ecology, and voila, post-infectious IBS. So what we're talking about here is a difference between a healthy gut microbiome and consequential metabolic function and degradation of that microbiome and associated gastrointestinal health and metabolism. So here on the left side, you're looking at homeostasis in an ideal textbook kind of way. And in a homeostatic state in the gut, as you know, you've got a healthy ecology, a high degree of diversity across the microbial system. You have a mucus layer, which is an outer and inner layer of mucus that protects the gut mucosa. So in the outer layer, there's a higher preponderance of gut microbiota. In the inner layer, there's less, but there is some interesting bacteria living in there. And generally, this is associated with tight junction proteins in the gut itself, and also immune homeostasis. There are interactions between the gut microbes and the immune system that actually favour inflammatory balance, autoimmune balance, and a good state of health. Of course, in dysbiosis, we're looking at exactly the opposite of that. So there's often disruption to the ecology, including loss of diversity, and this ultimately results in the preponderance of certain species that are associated with pathogenic effects, and also the loss of mucosal health as well. So often the mucosal layer becomes degraded and dysfunctional. The proximal relationship of the bacteria to the mucosa become a bit closer, and that then interacts with the immune system, causing inflammation. So things like non-adherent E. coli get a bit closer and activate the immune system. It can also change the organization of the tight junction proteins in the mucosal layer, opening up what we call leaky gut or intestinal permeability. Then we have increased passage of the endotoxin through into the sort of portal vein, and we have all of a sudden metabolic endotoxemia, which is activating the local immune system, and voila, you have chronic low-grade inflammation and the potential to develop autoimmune type scenarios as well. So this is ultimately what dysbiosis is and what's going on. Our current and prevailing theory of why people develop dysbiosis is a lot like most chronic diseases, except we're just talking about bacteria. So like most chronic diseases, really it's the perfect storm of environmental factors and genetics that are driving this change in ecology. In particular, environmental factors that are known to impact the microbial ecosystem itself and the health of the gut and gut mucosa. So these are the key things that are driving this dysbiotic change. So the question we need to ask then is, well, what's changing it? Well, actually, that opens up a really big discussion, well beyond nutrition, because there are a lot of different things that we're beginning to understand within our environment that have a huge impact on our gut microbiota, including a lot of early life factors. So we know that how we're born, whether or not it's cesarean or a natural birth, has a big impact. Early contact in life with the mother, whether we're breastfed or bottle fed, also has an impact. Early life exposure to antibiotics and medications. Where we're born, of course. And then there are things like hereditary influence. We also know that genotype plays somewhat of a role, although it tends to be less so than the environment. And certainly gender seems to influence the microbiota. We know that age, there are shifts in the microbiome with age that are quite characteristic, as well as environmental factors, of which there are many. But some of the things that we know are particularly important are exposure to radiation, which is interesting, as well as chemical exposure has quite a pronounced effect. We'll look at some examples of that in the context of nutrition in a moment. And then further to that, we also know now that, or at least there's some evidence to suggest that physical activity might even shape the microbiota. So the group at the Elementary Centre in Cork were able to show that high levels of physical activity improve microbial diversity. And further to that, we also have psychological stress, which we've known for a little while has been able to increase changes in the gut microbiota. So physical and emotional stress, as well as just general health status. So our health probably impacts our gut microbiome, but also vice versa. And then finally, and what we're going to focus on, are dietary factors. So what within our diet is influencing the microbiome? So before we get into that specifically, I just want to highlight a few big things that change the microbiome, and that is firstly the first 1,000 days of life where the gut microbiome is established. I feel like I've sort of made that point. We also know that there are some rather esoteric things, including our geography and environment. So for example, this group were able to show that actually people shared common microbiota than with the people that they lived with. This is particularly on the skin, but fascinating. And they also found that there were shared microbiota across their pets as well. Weird. Another thing that's also been demonstrated is that there are shared, actually the diversity of the ecology that we live in influences the diversity of the ecology of our microbiome. So that it seems that environmental diversity is favourable to our microbiological diversity. Now that's interesting, because that takes us out of the gut and takes us to a worldview of dysbiosis as being not just a human isolated individual problem, but a problem that is related to massive environmental change, whether it's dietary, which is the focus of our discussion. Of course, there's massive upheaval that's occurring around the world at the moment in terms of dietary change, but there are other changes as well, like environmental degradation and pollution that could be influencing the gut microbiota. Now, that's an interesting view. Also, analogous to environmental degradation and its effects on climate instability, we also know that degradation of our gut microbiome, including loss of diversity of species, has an important effect as well. It seems to be one of the characteristic features of a healthy and unhealthy microbiome is that diversity or lack of. And lack of diversity has been correlated, as this group were able to show, with different diseases, but also metabolic markers, which is what you're looking at here. So there are high levels of adiposity, insulin resistance, more inflammatory mediators, as well as preponderance towards weight gain. Also, harrowingly, and this is the bad news, we've also seen evidence to suggest that maybe our microbiota are slowly becoming extinct because of these environmental changes. So this group have been doing some stellar work and have been able to demonstrate that actually microbiota do become extinct over time. And they performed this really interesting experiment where they fed over successive generations mice lower fiber diets, and were able to show that there was a loss of diversity in even species across generations. And what was fascinating was they also were able to show that in early stages of this loss of species and diversity, feeding prebiotics was able to, at least at some level, almost adequately restore that. But through subsequent generations, it was very difficult to restore the health of that microbiome. So what does this mean for humans? We don't really know yet, but what it's suggesting is that there may have been progressive loss and destruction of our microbial ecosystems over time, and that could in part explain why it's sometimes difficult to recover, people to states of health. So I think that's really interesting. It's also very difficult to sort of re-establish and recolonize the gut. Really, one of the sort of more promising ways that we have that is emerging as a solution for that is this fecal microbial transplantation, which kind of acts as a reset almost. So let's jump back to the field of nutrition. And what I'd like to do is highlight just a few things. I'm just going to take a top-level scan through some hand-picked sort of studies through the literature that show some of the things that we know are disrupting our microbiome. So these include Western dietary patterns, and of course on the flip side of all of these, there are alternate sides to this. So Western dietary patterns we know are important. Excessive energy intakes are really key. We also know that alcohol can be quite disruptive, but the flip side of that is in moderation it may even be positive. And then fats, depending on the type and volume we're eating, has an influence. Refined carbohydrates, artificial additives, food contaminants, as well as sugar-containing drinks, low plant diversity, high heat processed foods, which we'll look at, as well as sterile foods, which is an interesting topic as well. So let's look at a few examples of how this could be impacting the gut microbiome. Well, firstly, looking at traditional versus Western diets, we know from quite a few reports now that traditional populations, including around the Amazon and Africa, versus North Americans and Europeans, tend to have a richer microbial ecosystem and have a better metabolic function of that ecosystem as well in terms of production of things like short-chain fatty acids. So this is an example where we're looking at changes in those ecology and the metabolic function in Italian kids versus African. Of course, there are other things at play here. There's exposure to soil and microbes and parasitic infections and perhaps less antibiotics and things like that. But it's interesting nonetheless to see that these differences are quite striking. We also know from more experimental work that there is something going on here. It could be a lot down to diet. So in studies like this, where you look at high-fat, high-sugar feeding studies, in this case we're looking at mice, I believe, what you typically see is that there's a dysbiotic scenario is set up pretty quickly, with a Western type diet model. And what happens is you get increased levels of these pro-inflammatory bacteria. One of the better studied is this, adherent E. coli, which then is associated with a breakdown of the mucus layer, so you get more of these bad bugs getting closer to the gut epithelium and starting to induce inflammation and inflammatory changes. So diet alone seems to be doing this. Another thing that's going on is perhaps excessive exposure to alcohol. So we've known for a long time that one of the reasons why alcoholics, which is an extreme scenario, develop fatty liver disease is because of increased passage of endotoxin through the gut, portal vein hits the liver, Kupffer cells get activated, voila, you have cirrhosis and eventually other serious problems. So essentially the effects of alcohol do change the gut ecology and do cause leaky gut and high levels of endotoxin in the body. This is also true for more modest consumption. So even a single bout of binge drinking, a single binge on alcohol will elevate significantly endotoxin in your blood for about 24 hours. So even, less extreme scenarios will have an effect on the gut ecology and circulating endotoxin. Another thing that we know mostly from experimental studies, there's not been a lot of human work yet, is that the type of fats that we're eating probably has an influence. We know that they actually can shift the gut microbial ecology, so they have pre and probiotic effects or perhaps even pathogenic effects or shifting the microbial ecosystem depending on the type of fats that we're eating. So as you would expect, the general sort of idea is that more refined omega-6 rich vegetable oils tend to be negative, whereas omega-3 fats, which are associated with more positive health outcomes, tend to have a more positive effect on the gut ecology. So that's kind of interesting. What's fascinating too, actually, and what this study was able to demonstrate is that it may not be just the fats locally changing the gut microbiota when we consume them, but actually the balance of fats in our tissues. So what they did is they manipulated in a transgenetic model the tissue levels of fats. So they changed them from an omega-3 rich mouse to an omega-3 rich mouse to omega-6 rich mouse and vice versa. And basically what they were able to show is that as you change the makeup of fats in your body, your gut ecology changes. So maybe your sort of omega-3 status, not just its acute effects on the gastrointestinal ecology, is really important, is what this is suggesting. So they found here that this shift in omega-3 to 6 ratio activated intestinal alkaline phosphatase, which is an enzyme that's involved in inflammatory regulation and detoxification of endotoxin. That changed the gut ecology, and this then influenced the passage of lipopolysaccharide or endotoxin, which of course binds to the toll-like 4 receptor on the surface of the cell and activates this inflammatory response. So that all shifted depending on the body level of these different fats. So that's interesting. Another thing that we think is going on is the type of additives that we're consuming are having an effect. And I found this particularly interesting because it's an area that is relatively new in terms of investigation, but we're seeing some striking things. At least in experimental studies, there's hard to delineate in humans, but it's fascinating, but also scary. So what we've seen are things like glucose salt emulsifiers and organic solvents, as well as gluten, which will be talked about later, and all this stuff here, microbial transglutaminase, which is used as a processing additive in a lot of foods, but it's not on the label. By law, you don't have to talk about it. It's in a lot of different foods, and also even nanoparticles may be influencing the gut ecology and gut immune function and even intestinal permeability. Now, some of the better examples of this are artificial sweeteners. So it was shown in the journal Nature, I'm sure you caught this one, but in the journal Nature that one of the ways that these artificial sweeteners are disrupting blood glucose homeostasis is likely due to their negative effects on the gut microbial ecosystem. So we've known for a while that even if you drink artificially sweetened drinks, you're probably still going to increase your risk for diabetes. So this is one of the reasons it seems why, one of the mechanisms, is that these sweeteners are changing the gut ecology that then disrupts blood glucose homeostasis and potentially increasing risk. Another thing that's been highlighted recently as a problem, also in Nature, but 2015, is the impact of these emulsifiers in our diet on our gut ecology. So it turns out emulsifiers basically sweep the gut lining. as Patrice Kenny put it. So what's happening here is that we're exposed to a very high level of emulsifiers in our diet these days. They're in everything that comes in a packet pretty much and has fat. So emulsifiers are used to homogenize the fat into the food, and as you know, they emulsify fats in most processed foods. So what this group decided to do is, say, what are the effects of these emulsifiers in both on the gut ecology but also on metabolism? So they took a couple of mouse models and they basically fed them a very low dose of emulsifiers that mimicked human exposure, so it wasn't excessive amounts. They gave them either the control or diets containing a few different emulsifiers, polysorbate 80, one I'm more familiar with. So what you see here is in the control, they retained a healthy mucus layer, which is stained for green here. Their gut ecology was nice and healthy here. That's the epithelium stained for purple. And the red here, you've got E. coli. So this is non-adherent E. coli, and it's potentially a problem if you don't have a mucus layer. When they consumed the emulsifiers, the mucus layer broke down. The E. coli was able to infiltrate into that mucus layer, and in fact it started to directly interact with the gut mucosal epithelium. It induced inflammatory changes. And in the general mouse model, it started to induce what we would characterize as being diabetes, obesity, and cardiovascular disease. They also found that in a mouse model that was genetically prone to inflammatory bowel disease, it triggered the inflammatory bowel disease. So this is really interesting. Of course, it's just mechanistic, but it's suggesting that perhaps some of these things in our food that we thought were benign, like emulsifiers, are not, and they're having important physiological effects on our gut microbiome. The same is also true for maltodextrin. Maltodextrin is another food additive that is ubiquitous. And there's a group that have been looking at this, and they've found very similar effects. It tends to be associated with changes in the mucosal barrier, breaking down the colon, and is associated with gut inflammation and, a very similar sort of scenario. So maltodextrin, again, in a lot of foods and even dietary supplements. So beyond these additives, there are also things in foods that we haven't put in there necessarily on purpose, and these are the pollutants. So things like dioxins and PCBs, and, the list is endless of the stuff that our food is contaminated with. The question these days is not, is my patient toxic as they are? What am I going to do about it? Or is it contributing to their health? And in terms of pollutants' effects on the gut ecology, there seems to be something going on there. Interestingly, our gut ecology tends to have some interesting protective effects against these pollutants and toxins, but it also could be negatively influenced by them as well. As this group found here, there was an impact of dioxins on the gut microecology. In fact, it reduced diversity, as you can see here. At least, experimentally, there was this nice sort of dense network of bacteria, but after exposure that had eroded. And then this, for me, I don't really understand this even, but there is this idea out there that maybe there is low-dose antibiotic exposure in our food. I've dug into this and I can't work out whether or not that is real. But of course we know that antibiotics are used in food production. As far as I can tell, there are stringent controls and laws on this, particularly in the EU. However, they can be used illegally or they may not be monitored. So there may still be low-level exposure to these. I don't really know. And I also came across an interesting study where a group of people were given a Buddhist vegetarian sort of detox diet for 5 days, and they monitored antibiotic traces in their body. None of them had had previous medical exposure, and they found elevated levels that declined over time with the diet. How valid that is, I don't know, but it's suggesting that there may be background exposure in some people. Probably depends a lot on which country you live in and your geography and the quality of the food that you're eating. But anyway, we know that antibiotics negatively affect the gut, and certainly exposure to these medicines in general can have a huge impact, but it is also possible that low-dose exposure would have an impact as well. And then another thing that's going on in Western diets is we've lost diversity of food. So our ancestors, and I'm talking about about 8 to 10,000 years ago, I'm talking like pre-agriculturally, had a repertoire of about 500 to 1,000 plants that they were eating regularly. That's what we estimate hunter-gatherers were consuming regularly. They were seasonal, they were, organic, 500 to 1,000. The average in Western populations is about 24. So it's changed dramatically. That diversity has been lost, and the seasonality has been lost as well. That's gone. I can eat bananas in the winter here, no problem. So the lack of diversity is really important, because if your gut microbiome, a biomarker of health is diversity, one way to encourage that is to eat a diverse diet, because remember what the function of these bacteria are, is they live off different foods. So different foods will therefore encourage the growth of different species, because they all have specialist roles. Some are fermenters, some are starch degraders, some metabolize proteins. They're all quite different, some influenced by phytochemicals in foods. So eating a very diverse diet will encourage diversity within the microbiome, but this of course has been eroded. So what are you going to do about that? Well, you could eat a paleo diet. But actually a lot of the ways we think about modern paleo diets is a bit misconstrued. So really, if you want to eat a paleo diet, what you want to do is eat mostly plants, because it's a misnomer, unless you're an Inuit, that you're eating meat all the time. And you want to eat seasonally and a really wide range of plants, probably not a paleo brownie. And then, of course, it's not just what you're eating, but the way you're preparing it. And I've been looking for a study like this to be done for a while, because I have a sort of pet hate against fried and industrially cooked foods. Because when you fry and industrially cook foods, which is what a lot of people are eating these days, you create chemical adducts or byproducts that are not in home cooking. As Michael Pollan says, you don't want to let corporations cook your food for you, because they do weird things to it. They put weird chemicals in it, and they cook it at heats that you would never cook at. 300 to 400 degrees Celsius in an industrial oven, or they'll deep fry it or whatever. So what happens is, when you expose proteins and starches and fats in particular to these very high heats, they change, and you start to form what we call advanced glycation end products. And we know that these have really important physiological effects. If you put this side of the room on a fried food diet, whereas if you put this side of the room on the same food, but get them to stir fry it and steam it and slow cook it, they'll have lower oxidative stress and inflammation. And it's because you guys are absorbing the advanced glycation end products, and they activate the receptor for the advanced glycation end products that triggers inflammation and oxidative stress. Well, it turns out that these advanced glycation end products seem to impact the gut microbiota as well, as this group were very recently, this is March this year, were able to demonstrate. And then another thing is, beyond cooking, there's also cold heat, right, or fermentation. So it's the way we've been traditionally processing foods for a millennia, and that's been eroded as well. So this study was really interesting. It was an ethnobotanical sort of survey of Eastern European traditional fermented foods, and there's more than you can list there. There's hundreds of different types of traditional beverages and foods and way of preserving things that actually utilize bacteria, and these bacteria are then consumed, of course, and then they probably have important effects on the gut microbiome, although this is still relatively new area of investigation. But I think there's something to it. So this then leads us in, if we can see quite clearly that Western diets have an important impact on the gut microbiome, it leads us into inflammatory disease and its connection there. So we've induced dysbiosis by eating a highly processed, refined, industrially produced diet. And what happens is it shifts us from this healthy gut microenvironment to a state that becomes more dysbiotic and is associated with pathological inflammation. One of the characteristics of this is we see a breakdown in the mucus, and you see this degradation of the mucus layer. You start to see adhesion of bacteria there, and initiating inflammation within the gut itself. And one of the characteristics of this, as I've pointed out frequently, is that we have lack of diversity. So it seems to be a hallmark feature of this thing that we call dysbiosis. An interesting phenomenon that tends to be occurring in this sort of dysbiotic scenario is what we call a bloom of pathobionts. I just love that phrase. Sounds like something out of Star Trek. Look, Captain, it's a bloom of pathobionts. So basically, what we're talking about here is a proliferation and expansion of pathogenic or bacteria that are associated with pathogenic effects. So this pathobiont bloom is really interesting to look at as in terms of why it's developing and why it's associated with severe inflammatory disease, like inflammatory bowel disease and these extreme scenarios. What seems to be happening is that in a normal sort of healthy gut, if there's such a thing, you basically have a sort of balanced community, so you have this healthy, stable community. These ferment, food, starches and things, and you get these fermentation end products. And we've got a nice little system going on, a nice relationship. But what seems to be happening in something like inflammatory bowel disease is you have a lot of inflammation, right? Including in the gut mucosa. The gut is inflamed. For whatever reason, that's what we're looking at clinically. Now, whatever's triggered that, it doesn't really matter now because we're left with an inflammatory scenario. And what's happening is that there's a lot of inflammatory mediators being produced, and there's also a tremendous amount of oxidative stress being produced as well. So there's a lot of free radicals and free electrons being produced. Now, these free radicals and electrons start to bind with substances in the gut. So they're actually released into the gut, and they start to bind to form nitrosative substances. Now, what's interesting is that this flood of oxidative stress and nitrosative substances into the gut itself feeds the pathogenic bloom. So it's driven by the inflammation. So what happens then is that enables certain types of bacteria to proliferate rapidly and bloom. And we end up with a dysbiotic state. And that becomes a self-perpetuating cycle because these bacteria are associated with inflammation. So if you want to look at it in more detail, there it is. So you have a host inflammatory response down here that recruits inflammatory cells, which then produce inflammatory mediators and cytokines. Of course, one of the core components of an inflammatory defense system is to produce things like hydrogen peroxide and other sort of free radicals, to kill off, pathogens. These are then released into the gut itself, and they actually start to form these nitrosative substances, which then fuel the growth of dysbiotic bacteria, and you end up with dysbiosis. So the take home from this is that this inflammatory state is driving this pathogenic bloom. So one of the areas that's been studied in this area, or in this concept, is this particular proteobacteria bloom. And it's interesting, these have been observed all throughout the ecosystem. I just kind of like that. So I've thrown that in. But also in the GIT, they're actually more abundant in a biopsy than they are cultures. So they're perhaps not that easy to identify through the methods that we're using to assess gut ecology. But a balanced gut microbiota tends to keep these bacteria in check. However, one of the interesting features of these particular bacteria is that they're actually facultative anaerobes. So in contrast to an obligate anaerobe, they're able to thrive in both oxygen-rich and oxygen-depleted environments, so they're a little bit different. And given the right environment, including inflammation, they will thrive and bloom. So that's what we're looking at there. And there is some work to suggest that they have a preponderance in certain scenarios. So for example, in healthy individuals, you have a fairly low level of these bacteria, whereas in a neonate, while the gut ecology is still establishing, there's a higher level that tends to stabilize, of course. But then there are more pathogenic related scenarios like metabolic disorders as well as inflammatory bowel disease and even gastrointestinal cancers that are associated with a bloom of these noxious bacteria. So the take home from all of this, just bringing it back to real world terms, is that we think there's a connection between our gut bacteria and disease. is particularly chronic diseases. But the funny thing is the jury's still out. As we've just seen, there are bidirectional relationships that we don't really understand. Is it a consequence or a cause? It's not totally clear, I don't think. And probably it doesn't matter anyway. It seems to be an important mediator. And modifying the gut microbiome with things like probiotics is important. and it has important clinical effects. What we do know, and this is the best review on the state of the science that I've seen, if you want to bookmark it, is that there is an association with dysbiosis, whether it's metabolic or, species-related, and a whole host of very common disorders, including allergic disease and asthma, inflammatory bowel disease, obesity, other non-communicable diseases like heart disease and diabetes, and also, neurodevelopment in kids, some remarkable work that's coming out there, and also there may be a connection with mental health, of course. Well, there certainly is. So from all this, what I'm going to do is just take you through a conceptual idea of how we could be modifying the gut microbiota, because we've got about 10 minutes. So what I've done here is come up with a sort of framework that is how I think about what's going on here, and I think it will be useful as a sort of reference point. So I'll fill this in as we go. You've got the full image in your notes, but basically down here we have what we've kind of been talking about. So we have microbiome diversity, which is correlated with health. In an ideal scenario like that, we have low levels of inflammation both in the gut and systemically. And this, as you know, inflammation is one of the key drivers of metabolic disturbance. So if you have endotoxin moving into your bloodstream, it starts to disrupt, things like your metabolic regulation, including insulin resistance and, metabolism in general. It results in peripheral inflammation in fat cells and in the brain. The list goes on and on. But these are some of the core consequences of chronic low-grade inflammation is disruption of barrier function. You could have elevated endotoxin oxidative stress and metabolic health on the flip. So, what can we do about it? Well, there are a number of different dietary strategies that we could take that are probably going to depend a lot on the individual. So I don't think there is a magical diet for everyone. As you all know, it doesn't work like that. Everyone's very individual. And I think increasingly so in modern environments. We're a lot more sensitive to dietary changes than perhaps we ever have been. So modifying and personalising diets is becoming more and more important. So, what is driving, what could we potentially do to modify the gut microbiome? Well, one approach that's been explored is low refined carbohydrate and low sugar diets. In fact, they've been explored for over 100 years, well, almost 100 years. So there's a good history around the use of these diets on and off. They've been given different names. More recently, we talk about things like the specific carbohydrate diet, but they seem to modify the gut microbiome. Another approach, of course, is pre and probiotics, which are immensely powerful in terms of manipulating, metabolic health and improving patient outcomes. So that's something to consider. Not only dietary supplements, but also pre and probiotic foods, of course, would fall into this broad category. And then, of course, you can encourage high plant diversity. And this seems to have an impact as well. So that can improve, the microbiota and reduce inflammation. And, it's been used successfully to manage things like inflammatory bowel disease as an approach. Another thing that's being explored, of course, is the impact of polyphenol-rich diets. So we know that polyphenols alone, like just drinking a cup of green tea, will change your gut microbiome. And that's because polyphenols are metabolized by gut flora, so they're in a sense a prebiotic, but also they have antimicrobial and antibacterial effects, so they're also a natural mild antibiotic as well, which is fascinating. Another thing we can do is implement oligoantigenic diets. So that is identifying food allergies and sensitivities and personalising a diet based on that. So Tom O'Brien's going to hit that later for sure. And we're also going to talk, focus on organic approaches as well, because eating organic food minimises exposure to things like plasticizers, we hope, but certainly pesticides and organochlorines and persistent organic pollutants. And that could have an important impact. I think it's really valid. And also, as I mentioned, you could also focus on the way people are cooking food. So, transitioning them from high heat processed foods to things that are a little bit more traditionally cooked and eating more raw foods and foods that are cooked with cold heat or fermentation might be a nice approach as well. And that brings us to fermented foods. And certainly introducing more good traditional fermented foods back into people's diets, I think, would be potentially beneficial. Another approach that has been given little serious investigation is the effect of lower protein diets, because some people over-ferment protein into hydrogen sulfide, and that can cause symptoms similar to IBS. So for some people, actually modifying their protein intake could be really important if they have bacteria that are driving that fermentation and symptoms. So Peter Gibson recently published a really good review on that. If you want to dig into the literature on that, it's excellent from a clinical perspective. And then you also have intermittent fasting, which I love the science around this at the moment. It's, for years we've been told you should eat 3 times a day and have 2 snacks in between, and now that's all flipping on its head. I've been waiting for that to happen, because we've known that our ancestors, like hunter-gatherers, would normally only have 1 meal a day. So when did it change? Well, the Industrial Revolution and longer working hours and then food marketing all had a massive influence on the frequency of eating. And it may be that actually the volume and frequency of calories that we're eating also influences our microbiome. And I'll show you a little bit of evidence that suggests that in a second. So all these things are really important from a dietary perspective, but I actually think we're missing part of the picture here. What is also fundamental to changing the gut microbiome are the functional things that are going on inside our patients' bodies. If your patient has hypochlorhydria, they're going to have dysbiosis. If they have low gastrointestinal motility, that's one of the main causes of small intestinal bacterial overgrowth. If they have, IBS, that will change the gut ecology as well. And, it's not necessarily only a gut microbiota related problem. IBS, like most chronic diseases, is multifactorial. Maybe they also have high levels of inflammation, as we just seen. That inflammatory storm can cause a bloom of pathobionts and completely change the gut ecology. So actually, a big part of your win in terms of manipulating the microbiota towards a more healthful state is not only addressing the dietary factors that drive it, it's also exploring some of the functional causes within our physiology and within our digestive health that we could be changing the gut ecology. So it's a whole systems, whole person, functional medicine-based approach. That's the kind of lens that we're looking at. There's not a single, magic pill or quick fix. There you go. So there are a few things that we can do in terms of assessment, of course, when we're looking at the microbiome from a clinical perspective. A very simple thing to do, and I think by far the most powerful assessment technique of anything is just good quality patient assessment. So taking a good quality case history, looking at their health history, previous antibiotic use, how were they born, what environments have they lived in, what stress have they been under. All of these things will paint a picture. So their story. We also have dietary assessment, so that's going to give you a bit of a telltale pretty quickly based on what they're eating. Of course, you can also look at symptom scores. So look at IBS symptom scores or IBD symptom scores will give you a good indicator of the severity of dysbiosis. Often these are strongly related to dysbiosis in the gut. There's also the Bristol Stool Scale. I'm going to come back to that in a second because I think it's really fascinating, this recent publication. And you can use these traditional and functional tests that a lot of us are using as well, like stool tests, I think are really valid still and can give you a good idea on the microbiology, the parasitology, that kind of thing. They're still really great clinically. You can also look at antigen tests, and you can look at breath tests for small intestinal bacterial overgrowth. You can look at low-grade metabolic endotoxemia. Labs like Regeneris out here do the autoantibody test, so that's a great one. And you can also look at lipopolysaccharide binding protein. Another thing you can look at is response to treatment, of course. So if someone's responding well, to your intervention, it's possible that their dysbiosis is improving. Back on the stool scale, this cool study was published. I love this because it basically suggests that very sophisticated investigative techniques actually correlate with one we've all been using in practice for years. And what they were able to demonstrate just recently is that actually very strongly your score on a Bristol Stool Scale, which as you know is this sort of visual scale on either the firmness or looseness of the stool correlates very well with microbial diversity in certain species. So the short version of this is if you have a healthy stool, somewhere around the mid to the beginning of the scale, you have better diversity in your gut microecology, and you also have a preponderance of some of these really health-associated bacteria like the Archimedesia species, which live in the mucus and produce anti-inflammatory substances, are really an interesting one. But they seem to be very important for maintaining gastrointestinal and systemic health. So cool, huh? You can just do a stool scale visual test and that'll give you a good indicator on gut ecology. So then, we've got a few minutes to just flip through some of the dietary interventions that we know work, and I'm probably going to move fairly fast on this. But there are a few things that we know in the context of diet could be really powerful. One of them is transitioning to people to a more traditional diet. They did exactly that in this study. They took a group of Africans living in Africa, they took a group of North Americans living in North America, and they swapped their diets and vice versa, and looked at the effects on their gut ecology. What happened? Striking things happened. They actually changed the inflammatory mediators significantly. They also changed like precancerous cell markers. The whole gut ecology changed. The butyrate production shifted, and you can actually see evidence of that here. Here you're looking at the North Americans before, so there's a lot of inflammation, so these are macrophages in the mucosa here, and as they shifted to a more African, high-fiber, traditional diet, that just started to disappear. So the gut mucosa was changing, the inflammatory mediators were changing, so you can go a bit hunter-gatherer and it'll be good for you. They didn't eat brownies. So beyond this, there have also been other striking examples of how what we're eating can shift our gut microbiome, including this, which is like the archetypal study that's been done, where they took a group of people and switched them to completely plant or completely vegetable-based diets. So extreme, sure, but it gives us a really good idea on how the diet can impact the gut microbiome. So I'm not going to go into the core details here, but what they basically found was a few interesting things. Firstly, it shifted the total gut ecology dramatically within a day. There were huge effects, and there were really important effects on health markers. So for here, I'm just pulling out a few. They found that there were reductions in these bile acids in the gut, which we know are involved in the initiation of colon cancer, but also have an effect on the gut ecology. So that changed dramatically. They also found that that transition to a more animal-based diet versus vegetable-based diet resulted in the production of more potentially pathogenic bacteria, like Biophilia wadsworthia, which is associated with inflammatory bowel disease. Perhaps a high, meat, processed, fat, protein-rich diet devoid of plants, not that good for you. And then there are more subtle effects from more real-world interventions, and we've seen a couple of studies now looking at the effects of the traditional Mediterranean diet on gut ecology, suggesting that, again, there's, as you would expect, benefits. So that's important. And then, of course, if we're really like red wine, we can rationalize anything. And we know that one of the features of the traditional Mediterranean diet is small amounts of red wine with food, and it turns out that for the red wine lovers in the room, yes, that will shift your ecology and lower blood endotoxin in moderation. And I love olives as well. And actually, olives are a probiotic food, so they're full of lactobacilli. It's one of the ways that they're actually fermented before they're produced. And this recent study found that eating 12 olives a day, which contains about 20 million lactobacilli, was able to shift. They found traces of lactobacilli. It wasn't the most sophisticated, assessment tool, but they found evidence of a possible probiotic or prebiotic effect. But of course, they found important metabolic health effects as well. They actually found reductions in inflammation, oxidative stress, and they even started to see changes in body composition, which is interesting. But beyond justifying my love of olives and red wine, I want to get back to to some more real stuff. And this is, I said I'd talk briefly about intermittent fasting, but this is some evidence to suggest that actually moving to a lower or more modified caloric intake, and think in this case they dropped it by about 30%, their daily caloric intake, to induce weight loss, is that actually their gut diversity tended to head towards improvement. And this is particularly true for people that had low diversity. So that's interesting. And then, beyond fermented foods, what we're beginning to understand as our investigative techniques get more sophisticated is that actually a lot of things that we're eating are full of bacteria. Actually, if you just look at unsophisticated methods, about 30% of our diet is made up of fermented foods. A lot of things we don't even think of being as fermented are, like olives, for example. But there are also microbes hidden in our meals. So this group were able to show, because we have these methods now, that actually a bowl of salad is full of bacteria, and they're living in the leaves, even on a washed or sterile leaf. That's fascinating. Kind of freaks me out a little bit, but I think it's really cool. And then beyond improving the intake of fruits and vegetables and fermented foods and shifting to a more traditional diet, we also need to avoid sugars. And one of the reasons why is because the small intestinal bacteria love those. And that seems to encourage small intestinal bacterial overgrowth, which this group were able to show for the first time just recently, is that there is an association between high sugar, high refined carbohydrate intake and small intestinal overgrowth. So the whole yeast connection anti-candida diet has come full circle and now is starting to be justified. And this group were also able to show that a specific carbohydrate diet, which I feel personally is a little bit extreme, but interesting as an example, in a group of people with inflammatory bowel disease was able to improve microbial diversity. Now that's really interesting. Maybe modifying carbohydrate intake, in particular, getting away from easily digested starches that will encourage small intestinal bacterial overgrowth, not only would help reduce that, but also improve the overall gut ecology. And then I mentioned omega-3 versus omega-6 fatty acids before. We don't know a lot about how that plays out in humans, but I found this interesting case report. This is an N-of-1 study. So what can we take from this? I don't really know yet, but it's fascinating nonetheless. So I've just thrown this in here for interest. And what they found is that in this single individual, as they consumed a fish oil supplement, so just 400 milligrams of fish oils a day, they were able to document over time important changes in their gut microbiome. So of course this is N-of-1, so it's very difficult to work out what this really means, but they saw important shifts. So it may be that the type of fats that we're eating has an important influence on our gut ecology. What's also fascinating is that they found potentially that this would help explain why omega-3 fatty acids have shown very little benefit in Crohn's disease. So they saw a bacterial imbalance that was associated with Crohn's that was actually encouraged by fish oils, which is fascinating. And certainly it doesn't seem to work that well for Crohn's when you look at the literature. In ulcerative colitis, much better. And then where this is all arriving now is that this idea that we could actually manipulate the overall diet in a way that would encourage important changes in our gut microbiome, that would favour better metabolic regulation, better diversity, and overall metabolic health. And only very recently are these types of interventions starting to be really seriously considered. Like this one here, which I think is one of the best examples, where they actually devised a microbiome-targeted dietary intervention with the aim specifically of reducing circulating endotoxin and improving metabolic health. So this diet revolved around, a diversity of plant foods and prebiotics, and guess what? It worked. As they started consuming this microbiotic-targeted intervention, the lipopolysaccharide in the blood was reducing, the metabolic markers were improving, and their overall health was getting better. So with this in mind, I've also worked up a number of the concepts that we've reviewed here for a paper which is about nutritional regulation of metabolic endotoxemia, which has been accepted and will be published soon, I hope. So where we've gone with all of this is that there are a diverse range of environmental factors out there. that are influencing the gut microbiome, including early life factors, genetics, but what really concerns us from a clinical perspective are the things within our control, or the sphere of control of the individual, and these include, in particular, dietary factors. As we've seen, they have a massive impact on our gut microecology in a way that can shift the microbiome in a healthy direction, improve our metabolism, and improve our health. So what we've explored is a number of different dietary factors up here that we know, or approaches that we know could have positive effects on the gut microbiome. But what I've also left you with is the idea that actually it's not just about diet, it's also about function. And actually improving gastrointestinal function and physical function of the individual will have huge wins for improving their gut microbial ecosystem and their health as well. So I want to leave you with the idea that when we're talking about the gut microbiome, we're not just talking about individuals, we're talking about their connection to their physiology, because their physiology impacts that. We're not talking about just their bacteria. We're also talking about their diet. So we step out of the physiology and look at dietary factors that influence that. But we can also see, as we saw before, things like planetary diversity influence the gut microbiome. So there's a big picture here. This is not just a human health issue, it's an environmental issue. Thank you.
Thank you, Ben.
Thanks.
Thank you, everyone. I hope that you enjoyed that talk. And we now have a break until 11:40. So if you can make yourself available upstairs and network and enjoy the products that you've got in there as well. And see you back at 11:40. Thank you. We've got the questions and answers.